we are going to start with our new topic, that is fermentation. I'm Dr. Moina Akhtar. I'm a professor in Dr. M. A. Kazi Institute of Chemistry, University of St. Jamshedpur. So fermentation is basically of three types. What are those three types? It's lactic acid fermentation, ethanol fermentation, and acetic acid fermentation. So we are going to discuss each and one by one, right? So in today's video, we'll be talking about fermentation. It's a metabolic process of converting carbohydrates to acids or alcohol. This process occurs in microorganisms such as bacteria or fungi. Now, yeast, that is fungi, which is a type of fungus, converts sugar to alcohols, whereas bacteria convert sugar to lactic acid to obtain the energy. So now, we have the two types of fermentations, which are more important. So let's start with our topic. Now this is the table of contents which we are going to discuss today. That is introduction of fermentation, then definition of fermentation, then its history, then few examples, then the main topic, that is ethanol fermentation, and the facts regarding fermentation. Introduction to fermentation. Well, as I just said, it's a metabolic process that converts sugar to acids, gases, and alcohol. It occurs in yeast and bacteria and also in oxygen-starved, that is deficient muscle cell, as in the case of lactic acid fermentation. Fermentation, it's a chemical process by which molecules such as glucose are broken anaerobically. Now, these are the terms: anaerobic, aerobic. Aerobic is something to do with oxygen, that is in the presence of oxygen. Anaerobic, that is in the absence of oxygen. So the study of fermentation is also called zymology. Now let's start with the definition of fermentation. It's the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts produced by microorganisms like mold, yeast, and bacteria. Enzymes act by hydrolysis. It's a process of breaking down or pre-digesting complex organic molecules to form smaller and, in the case of foods, more easily digestible compounds and nutrients. History of fermentation. The term ferment comes from the Latin word fervere, which means to boil. So fermentation was described by late 14th century alchemists, but not in the modern sense. The chemical process of fermentation became a subject of scientific investigation about the year 1600. Well, it's a natural process. People applied fermentation to make products such as wine, meat, cheese, and beer, long before the biochemical processes were even understood. In 1850s and 1860s, Louis Pasteur became the first zymologist or scientist to study fermentation when he demonstrated fermentation was caused by living cells. However, Pasteur was unsuccessful in his attempts to extract the enzyme responsible for the fermentation from yeast cells. In 1897, German chemist Adolf Buchner ground yeast, extracted fluid from them, and found the liquid could ferment a sugar solution. Buchner's experiment is considered the beginning of the science of biochemistry, earning him the 1907 Nobel Prize in Chemistry. Examples of products formed by fermentation. Now, most people are aware of food and beverages that are fermentation products, but may not realize its important industrial products results from fermentation. Now, the examples are beer, wine, yogurt, cheese, certain sour foods containing lactic acid, including sauerkraut, kimchi, and pepperoni, bread leavening by yeast, sewage treatment, some industrial and alcohol production such as for biofuels, hydrogen gas. Now let's start with the ethanol fermentation. As I just told you, it occurs in yeast and some bacteria and follows anaerobic pathway. Let's start. Yeast and certain bacteria form or perform ethanol fermentation, whereas pyruvate from glucose metabolism is broken into ethanol and carbon dioxide. The net chemical reaction for the production of ethanol from glucose is as follows. Glucose, it gives 2 molecules of ethanol plus carbon dioxide. Ethanol fermentation has used the production of beer, wine, and bread. It's worth noting that fermentation in the presence of high levels of pectins results in the production of small amounts of methanol, which is very toxic when it's consumed. And let's start with this schematic diagram. Now in here, glucose is converted to the 2 molecules of pyruvate. And 2 molecules here we have of ADP, that is adenosine diphosphate, in addition to 2 molecules of phosphate, which forms 2 ATP molecules, that is adenosine triphosphate. So this process here releases 2 molecules of ATP at the same time. NAD plus is converted to NADH. And these are electron carriers. Fermentation facts. Fermentation is an anaerobic process, meaning it does not require oxygen in order to occur. However, even when oxygen is abundant, yeast cells prefer fermentation to aerobic respiration, provided a sufficient supply of sugar is available. Fermentation occurs in the digestive system of humans and other animals. In a rare medical condition called gut fermentation syndrome or autobrevere syndrome, fermentation in the human digestive tract leads to intoxication by ethanol production. Fermentation occurs in human muscle cells. Muscles can expend ATP faster than oxygen can be supplied even. In this situation, ATP is produced by glycolysis, which does not use oxygen. Although fermentation is a common pathway, it is not the only method used by organisms to obtain energy anaerobically. Some systems use sulfate as the final electron acceptor in the electron transport chain.